In this video, I'm going to explain shortly about nature of bond linking monomers in a polymer, including carbohydrate, protein, nucleic acid, and lipid molecules. In a polysaccharide, the individual monosaccharides are linked by a glycosidic linkage. And this bond is formed by the dehydration, or you can consider one molecule of water is removed. So one molecule of water is removed so that glycosidic linkage is produced in between two monomers. I have given here maltose as the disaccharide where two alpha D glucose present in between two alpha D glucose alpha glycosidic linkage present. Whereas I have given another disaccharide, that is cellobiose, which is composed of two molecules of beta D glucose. Therefore, in between these two glucose molecules, beta glycosidic link is present. So maltose and cellobiose are disaccharides. So similar way for the polysaccharide, n number of glucose present in case of starch if you consider. So in that case, in starch molecule, alpha glycosidic bond present because it is composed of repeating units of alpha D glucose. Whereas if you consider about the cellulose, it is composed of repeating units of beta D glucose and therefore beta glycosidic bond present. So you can consider glycosidic linkage two types, alpha and beta. And sometimes you will find in this case you are getting that 1,4 alpha 1,4 linkage present, but somewhere you will find alpha 1,6 bond also present, especially if you consider about the glycogen and starch molecule. Along with the alpha 1,4 linkage, alpha 1,6 linkage also present. In a protein molecule, amino acids are linked by peptide bond, which is formed when carboxylic group, that means COOH group of one amino acid reacts with amino group of nearby next amino acid with the elimination of water moiety. So this is also dehydration process. In this process, water is removed, H2O is removed, so that there will be formation of peptide bond. Peptide bond means this is CONH bond. Okay, so CONH bond is present in between two amino acids. Phosphodiester bond is formed in between two nucleotides by releasing water molecule. So that means it is a dehydration or condensation process. In a nucleic acid, that means in DNA and RNA, a phosphate moiety links the 3' carbon of the sugar of the one nucleotide to the 5' carbon of sugar of the succeeding nucleotide. The bond between phosphate and hydroxyl group of sugar is an ester bond. So you can see here, this is the hydroxyl group and this is phosphate group. So bond between phosphate and hydroxyl group is known as the ester bond. But you can see here two ester bonds present. This is one ester bond and this is another ester bond. So here in the second picture you can see this is one ester bond and this is another ester bond. So therefore, it is known as the phosphodiester bond, and phosphodiester bond is formed in between 3' and 5' position. So this bond is known as the 3' 5' phosphodiester bond. In case of lipid, ester bond is formed in between glycerol and fatty acid. In this case also you can see uh, water is released here. This is water. So here this is also water released, and due to that process there will be formation of ester bonds. So in glycerol and fatty acid, in between that three ester bonds possible, and either these fatty acids are same fatty acids or it may be different fatty acids. So in triglyceride, ester bonds are present. If you think this video is helpful for you, kindly like, share and subscribe this channel.